I am going to switch to the document camera right now because I did want to make that one final point that we didn't get to in the previous lecture about the different types of gene expression, the differential gene expression. So we talked about the promoter region having the response elements and all the other machinery that's there, for example, the TATA box for the binding of the RNA polymerase that is responsible for producing the RNA strand from the antisense DNA strand of the gene. There are many different types of response elements that are important in differential gene expression regulation. And one of the most important ways of thinking about it are the different types of gene expression per cell, per tissue, organ. It's mostly cell and tissue that we would be thinking about. So there's cell-specific gene expression. So to use the pituitary as an example, why does only one type of pituitary cell, for example, the somatotrope, or tropic cell, express the gene for growth hormone? And so other cells in our body, unless it's a tumor type of a cell that's aberrantly expressing that gene, don't express the gene for growth hormone. It's only in the pituitary. So that description reminds us of a different type of specific gene expression, and that's tissue-specific gene expression. Those aren't smiley faces, those are to bring gene expression down there. So I said, why is it that only cells in the pituitary express the gene for growth hormone? Well, that's because there are tissue-specific regulatory elements or response elements. The RE stands for both conveniently, so either regulatory element or response element. These would probably be more for the use of the term regulatory element than response element. Response element tells us that it is a portion of DNA in the promoter region controlling gene expression in response to some change in the cellular environment, internal, external, for the cell, internal to the organism, or external to the organism. So a wide variety of stimuli that can cause responses at the level of the gene. As a regulatory element, we'd think of more ongoing regulation such as this. So the example we just saw was the PIT-1 regulatory element on the promoter region of just about all genes specific for the pituitary function in the pituitary cells. Excuse me. I mean, general housekeeping genes are going to be, just as the term implies, expressed in nearly in all of the cells in our body. Maybe not all the same general housekeeping genes, but many of the same general housekeeping genes, and structural genes too. Think of the genes for the cytoskeleton, so in lipid synthesis, and so on and so forth. One final point about regulated gene expression is that there can be two types of regulation. And this is more thinking in the term of a response element. It's still regulation, but beyond the level of either express it for this tissue and cell, or don't express it for this tissue and cell, for an ongoing, moment-to-moment basis for many genes, there are two ways that they can be expressed and thereby regulated, most likely through response elements. There is constitutive gene expression and inducible gene expression. Constitutive means it's on, and all we're doing is modulating it. We'll increase sometimes, hold steady at a high level maybe, or decrease back to baseline levels, or maybe decrease below baseline levels, hold at a low baseline level, or come back. So that is constitutive expression, always on at a certain baseline level, and it is modulated, or regulated if you want to use that term, to increase, hold at a higher level, decrease back to baseline, homeostatic recovery through maybe a negative feedback mechanism, or be depressed to a lower level and held there and then brought back to baseline or higher. So all those variations of regulating, or I like the term modulate more so. I like to think of regulation as turning your light switch on and off in response to environmental changes, and modulation kind of as a rheostat where you can make the lights brighter or dimmer, and you control how fast you make them bright and how long they stay bright and how fast they might come back down to normal. We'll discuss that more as we go along. And that's the difference between functional anatomy and physiology on one hand, and then the molecular biology on another. It's a bit different. It's all considered as regulation if it has the capability of being changed. Or even if it has the capability of being off at all times and on at sometimes. Inducible is where the gene can be expressed so all of the regulatory elements are bound by the appropriate transcription factors for that particular cell in that particular tissue, so that we get tissue-specific gene expression and cell-specific gene expression, but the level of expression is kept at 0. So baseline expression equals 0, if there is such a thing, right? In other words, the gene product, the protein of these inducible genes under normal conditions, is undetectable. So by all present methodologies, the messenger RNA levels are undetectable. The proteins, the gene products, are undetectable as well. So there has to be some stimulus to increase the gene expression from a 0 baseline up to this level here. So there's no going below baseline in this case. You either increase to whatever level might seem to be appropriate, maybe hold it at any one of those levels, who knows, for a little while, and then we'll always come back to the baseline 0. Now that's normal inducible for switch signaling. So when something comes on for a period of time and then it goes away, then we have this time-associated presence and absence that is indicating something has changed. Now this might be a bit different than a pathological induction of a gene. As I said, a cancer cell that suddenly now is producing various pituitary hormones or other proteins and molecules because of a pathological induction of genes. Most of those that you would hear about in normal physiological conditions, if it's inducible, it has a 0 baseline, some stimulus causes its expression. Typically that expression is transient, and in some time frame different for each gene, it will return to 0. But by far, most of the genes that we hear about or talk about are constitutive. They're on all the time to some baseline level, and they're modulated above and below that baseline.